Okay, hello everyone, welcome back. So this is the week 6 of the NPTEL course Experimental Biochemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor Dev from the Department of Bioscience and Biotechnology, IIT Kharagpur. And I am Bias Mukherjee, a research scholar from Dr. Shomodev's lab. And in today's class, we are going to talk about different gel electrophoresis techniques. Right, so gel electrophoresis is a very simple technique by itself, but it has a lot of applications in the study of different biomolecules. Right, so in today's class, we are going to talk about SDS page, native page, 2D gel electrophoresis, and then agarose gel electrophoresis and other techniques. Right, we are going to solve a lot of questions in today's class, and if you have any doubts, you can ask me. Also, at the end of today's class, we are going to discuss one question from last class on which you had a doubt. So, we are going to talk about that. Okay, right. So, let's begin with today's class. Okay, let's learn about electrophoresis. Okay, give me a second. Yeah, so we'll talk about electrophoresis, right? So what is electrophoresis? Electrophoresis refers to the property of migration of charged particles in the presence of electric field. Okay, now this laboratory technique is used to separate macromolecules based on their charge, shape, size under the electric field. Now, the migration of negatively charged particles or anions towards the positively charged electrode or anode is termed as anaphoresis, right? And exactly the opposite behavior, which is migration of the cations towards the cathode, is called cataphoresis. Now, electrophoresis has a very wide application in separating and analyzing biomolecules such as proteins and nucleic acids. Okay, so we'll start with the physics behind electrophoresis. So what exactly happens in electrophoresis and why do these charged particles move, right? And how are we exploiting their behavior in under different charged electrodes for their study, right? So the velocity with which the charged particles migrate in a constant electric field is termed as electrophoretic mobility. Now, the rate of migration depends upon the intrinsic properties such as net charge, shape and size of the biomolecule and the external factors such as voltage gradient of the electric field and the resistance offered by the medium in the form of friction. Okay, now you can see the formula for electrophoretic mobility, which is equal to V divided by E. E is your electric field and V is the voltage, and that is also equal to Q divided by F. So Q is the charge and F is the friction. Okay, so now Ohm's law states that voltage across the conductor is directly proportional to the current flowing through it. We know about Ohm's law, we have studied that in school, right? And also a lot of you might have studied this in your college. So it tells you that this particular formula tells you about this particular formula that whenever a voltage is applied across two different places, current flows and there is a resistance that is offered. Now, under conditions of constant current, velocity is directly proportional to current that we can see from this equation of Ohm's law. The velocity of the molecules is maintained but heat is generated, right? And that is largely because of resistance. So heat is generated under constant voltage conditions. The velocity slows, but no additional heat is generated during the run. So under constant power conditions, the velocity slows, but heating is kept constant. So now we have to use these these formulae to finally decide what voltage I want for the electrophoretic process that I'm going to perform and whether that is going to be detrimental to my proteins or DNA, because if there is a lot of heat that is generated, then the proteins and the DNA might get denatured, right? So then we will have to maintain the voltage. Okay, so we will go to the next part, which is what exactly happens in electrophoresis. So see, the mobility is a fundamental property of this macromolecule and its value depends upon the magnitude of its charge, its molecular weight and its tertiary or quaternary structure, that is its shape, right? So because most biopolymers such as proteins and nucleic acids, they are charged, they can be separated and quantified by electrophoretic methods. Now, the electrophoretic mobility of a particle is defined as a fundamental characteristic of that molecule under given conditions of support medium like pH, ionic strength, and temperature. The electrophoretic mobility is usually reported in units of velocity per unit of electric field. Okay, so right, so in that case, this equation would become velocity and this would be your electric field. Okay, so okay, you have a question? Yes, please ask. If you have any question, you can ask.
Ma'am, in past few days, I have been learning SDS for protein separation, and but happens that my protein of interest
Yes.
and diaphragm both get merged.
Can you tell me once
Like,
again? I did not understand. Tell me once again. What is your question?
ma'am, in past few days, I have been isolating proteins,
Hmm.
separation of proteins, and when I'm running the gel, diaphragm used to merge with the protein of interest. My protein of interest is around 38 kd, but it
38 kg. Diaphragm merges with your protein of interest.
yes, ma'am. Could you help me to troubleshoot that?
You can
I'm
write in
not sure.
research gate. Okay, you can write in research gate. You can check whether the Lamely buffer that you're making, whether it is proper or not. Okay, and the different gel compositions and all you can check. So maybe you need a lower percentage gel or higher percentage gel, that you can check. So how to do it? One of the ways is you write in research gate. Okay, so whatever is your problem, you write there and take a photo of that, whatever issue you're facing, you take a photo of that and you paste it. There will be a lot of experts all around the world. A lot of people might have even faced this problem that you're facing. They'll be the best people to answer, okay, to help you out. Because I have never seen a protein which is 38 kDa and it is running so low that the diaphragm is merging. So it is very new to me also. So I'm not sure exactly what is happening. Okay,
Sure, ma'am.
yeah, you have to possibly optimize the gel percentage, then the Lamely buffer composition that you're using, all these things. Okay, and see whether the protein requires any special kind of treatment with any other separate chemical or not. So all that you can check. Whether it is
Sure, ma'am.
breaking.
We'll check.
Okay, so in the if you're running the ladder, are you seeing that it is the molecular weight of the protein is corresponding to 38 kDa, or is it running very low because of some kind of cleavage that is happening in the protein?
Ma'am, actually, rest of the proteins which are supposed to be present in the band, less than 38 kd are not in the gel. They have passed through.
Okay,
Yes.
okay. So that means it is running very fast. So that means you need a higher percentage gel, I would say, because then the pore size would be smaller and then your protein will be resolved better. So what percentage gel are you running?
I guess 12%. Not sure.
12%, yeah, but then for 12% it shouldn't happen that way. You can check. Okay, so in ResearchGate you can post this question. So in 12% you should be able to see, and 38 kDa should come in the middle of the gel if it's 12%, right? So you can check that.
Sure, ma'am.
Okay, so let's continue. Since you are already facing this problem, so the other students will be able to relate to today's topic very well then. Okay, so yeah, that is how we have to optimize a lot of experiments in the lab, and that is what research is, and that is the beauty of research and also the difficulty of research, right? Okay, so we'll carry on and see what is happening. So there are two different electrodes. Since our DNA or protein, whatever we are using, that is negatively charged, so we start with the cathode at the side of the cathode and then it runs towards the anode, right? And we place it in that way. Then what happens is you run it for a particular duration. You set the voltage at around, if it's agarose gel, the voltage is normally 80 volts. And if it is protein gel, the voltage is normally 80 volts, 80 to 100 volts for stacking, and then 100 to 120 volts for resolving. And then what happens is you take out the gel and you view it. So we will see all of these different procedures, exactly what is done, how it is done in today's class. And based upon the size, we see that the separation has happened and all the larger bands are closer to the cathode that we had started from, and all the smaller ones are closer to the anode towards which the protein was traveling. Okay. Now, there are different types of electrophoresis. For example, there is gel electrophoresis, and under gel electrophoresis comes agarose gel electrophoresis, SDS page, native page, pulse field. There are different types of techniques, isoelectric focusing that we are going to talk about, 2D gel electrophoresis, all of these techniques. Then in zone electrophoresis, so there are different kinds of electrophoresis used for different purposes. You can read about them if you are interested. Okay, so there is this full table, full chart given for your understanding. Right. So we will talk about capillary electrophoresis in very brief, and then we will move on to the gel electrophoresis that we are going to discuss in details today. Okay. So in capillary electrophoresis, what happens is this separates charged molecules in a narrow capillary tube under the influence of an electric field. Okay, with smaller and more charged molecules moving faster through the capillary. So it is used for DNA sequencing, protein analysis, and the separation of small molecules like ions and amino acids, offering very high resolution and efficient separation in the compact system. So this is basically used for very small molecules, right? So now we will move on to the other gel electrophoresis techniques. For example, isoelectric focusing. Now this is a very important technique. We have a lot of MCQs on this in the upcoming slides. Okay, so what happens in isoelectric focusing? So isoelectric focusing separates proteins based on their isoelectric point. Now you remember isoelectric point, it is the pH at which the protein is having what? Completely neutral charge.
Zero
Right?
charge.
Yes. Yes, correct. Very good. Yeah.
Yes, I was just saying zero charge.
Zero charge. Yes, right. Very good. So where the proteins, and what happens is when they are having zero charge, that means they will neither migrate towards the cathode nor the anode. So the protein will stop there on that gel. Okay, so isoelectric focusing is a horizontal technique. It happens horizontally and the protein stops at the place where its PI is. Then what happens is this is widely used in proteomics to achieve high resolution of separation of proteins for subsequent analysis, such as identifying protein isoforms and purifying specific proteins. Okay, so what happens in isoforms is once this pH-wise separation happens, then you subject that to normal SDS page. So then what happens? The protein migrates again vertically, and based on their different molecular weight, they are going to get separated. Now there are different proteins that are having different kinds of post-translational modifications. So let's say that one particular protein is having some phosphorylation in it. So phosphorylation is giving it a different charge, right? It is giving it more negative charge. But then phosphoryl group is very tiny, so it is not changing the mass of the protein as such. So then what happens is at the SDS level, both the protein isoforms will be running at the same uh, like position, but at isoelectric focusing one will be slightly apart from one another. So then you will realize that that is a phosphorylated protein. Now when we come to glycosylation, glycosylation means a huge sugar moiety is added onto the protein. Then what is happening? The charge of the protein is at least the PI of the protein is not getting affected as such, but then the your mass of the protein is getting altered largely, and because of that we will see that there is this difference, right? So charge of the protein is getting affected slightly, but the mass is majorly getting affected. So you can see that in this SDS page gel after isoelectric focusing, this is one protein band and this is another protein band. So this one is glycosylated and this one is not glycosylated. Okay, so that is how we can learn about different isomers. Now what happens in gel electrophoresis. So whatever we talked about, we will just look at that once again. So there are these wells. Into the wells we put in the protein or DNA sample. So this is shown in case of agarose gel electrophoresis, which is a horizontal technique, right? So the gel is put, there is this buffer tank, power source, and the DNA travels, right? So DNA ladder is put to see which is the corresponding position of the DNA, and corresponding to that we have the ladder and the molecular weights, and we can say that okay, so this particular DNA is corresponding to this molecular weight. Now another thing comes in, that is the shape of the DNA. Okay, so we will look at that also in details, that even if the mass of two DNA are exactly the same, they can run differently depending upon the shape. That is called its topology. So we'll talk about that in the upcoming slides. Okay, now we will go through this. Okay, so gel electrophoresis separates biomolecules based on their charge by mass ratio. So when the electric field is applied, charged molecules such as DNA, RNA and proteins, they move through the pores in the gel matrix. The smaller proteins travel faster and the farthest, and the larger ones travel slow and late. Okay, so this technique is widely used in molecular biology to analyze the size of DNA fragments, RNA and protein. It plays a crucial role in applications such as DNA fingerprinting, genetic research, protein characterization, etc. Now, for proteins, now why can electrophoresis be applied on proteins? That is because proteins have these free termini, right? So they are having these termini, the negatively charged C terminus and positively charged N terminus, plus the side chains are there. Side chains can have different charge at different pH, because of which you can see that other than their isoelectric point or the pH at which the net charge is 0, proteins are charged molecules, and therefore they undergo migration towards the oppositely charged electrodes in an electric field. So this is the principle of gel electrophoresis and is used to separate protein molecules with different charge by mass ratio. Now DNA and RNA, it is having overall negative charge. Okay, both are having overall negative charge and they are uniformly charged, and they always migrate towards the anode or the positive electrode, and this migration depends upon the size and shape of the nucleic acid only. Okay, so now we will look at the principle behind SDS page. So a lot of questions can be expected from SDS page because this is a very basic technique, a very fundamental technique. Okay, so it's very important that you understand this properly. Now see, electrophoresis using gels made from a polymer of acrylamide cross-linked with bisacrylamide in the ratio 29:1. So this 29:1, this is actually 30% gel electrophoresis. So this is the normal one that is used in the lab. Okay, so this is referred to as polyacrylamide gel electrophoresis or PAGE. The gels are chemically inert and stable, and the copolymerization of acrylamide monomers. So acrylamide forms chains, bisacrylamide forms these connection between the chains. It is forming cross links between the chains. Okay, so cross linking reagent is N,N-methyl bisacrylamide that gives a transparent clear gel with pores. Okay, so all the structures are given to you. Now two more components are introduced in this slide. They are TEMET and APS. So APS is ammonium persulfate. So what is happening? This polymerization process, so the polymerization of acrylamide and bisacrylamide. So this process is a free radical catalysis. Okay, and it is initiated by APS and it is catalyzed by TEMET. So TEMET speeds up the reaction, but even without TEMET, this reaction is possible, but it will take hours then. Okay, so even like a lot of hours it will take. So TEMET speeds up the decomposition of the persulfate ion to give a free radical. The reaction is shown here for your reference. Now we will go to a deeper understanding of this. So you can see in this figure that these polyacrylamide chains are there and they are cross-linked by bisacrylamide. Okay, so the overall mobility of the protein samples in the polyacrylamide gel is proportional to the pore size, which is a function of the concentration of acrylamide as well as that of bisaccharidamide. So how big the pore is going to be, that depends upon the composition of your gel. And the pore size decreases with an increase in the acrylamide content. Okay, so as the percentage of gel increases, the pore size will decrease. So the smaller molecular weight proteins will get resolved better. Okay, so based on the molecular weight of the proteins to be analyzed, the gels of varying acrylamide content, hence different pore size, are used. So here it is given that for what percentage of gel, what molecular range protein is ideal to see. So you can see for 5 to 12%, if the molecular weight is 20 kDa to 150 kDa, it is ideal. For 10 to 15% gel, molecular weight of proteins 10 kDa to 80 kDa is ideal. Okay, for more than 15, if the molecular weight of the protein is less than 15 kDa. Okay, so now we have types of page. So polyacrylamide gel electrophoresis is normally of two different types. One is SDS page and the other one is native page. So what is the difference between two? So page can be performed in reducing conditions with an anionic detergent or SDS, or non-reducing conditions. Okay, in native or non-denaturing gel electrophoresis, SDS is not used and the proteins retain their native structure. So if you want to study a particular protein and its interaction with, let's say, DNA, then the experiment that you are going to do and the type of gel that you're using is native gel, and the experiment is known as EMSA, electrophoretic mobility shift assay. So the protein is there and the DNA is added, and as the concentration is varied, we see that the complex is formed and the mobility of the complex is tracked. Okay, so native page, in native page, the biological function of the protein is retained because the biological structure of the protein is retained. But in SDS page, what happens is a lot of SDS is added and the protein is denatured. It is completely linearized, opened up, and then SDS molecules go and coat the protein, so the entire protein becomes negatively charged. In the native gel, the protein is migrating depending upon its own charge. Okay, so now see. SDS is not used and the proteins retain the native structure and enzymatic activity. Here the separation of the protein is influenced by net charge, shape and size. But in SDS page, the separation is only based on the size of the protein. Okay, we rule out all the other factors. So where the SDS strongly binds to the protein samples and impart a uniform negative charge, so that the separation is purely based on the differences in mass, and SDS binds to the hydrophobic regions in the protein and it unfolds the protein. Okay, so it unfolds the protein and linearizes it by binding at intervals of every 2 residues. So remember this is a very important question. How many SDS molecules will bind if let's say there are n number of residues in a protein. So remember that one SDS molecule binds after every 2 residues. Okay, so the reducing agents beta mercaptoethanol and dithiotrietol or DTT, these are used to break the disulfide linkages if they are there in the protein, and in so the gel monomers are seen as distinct. In the gel, the monomers of the proteins are seen as distinct bands corresponding to their specific molecular weight. Okay, right. So here is the principle of SDS page that you can see very nicely. Okay, so when you are denaturing the protein, heat is applied. Heat is helping to denature the protein, and once the protein is denatured, you have beta mercaptoethanol which is added. It breaks the disulfide linkages. SDS is coating the protein completely. It is also assisting in denaturation of the protein because this is a detergent, right? And SDS is coating it and giving it negative charge so that it runs uniformly in the SDS page. Okay, so now the principle of SDS is once again shown here. So this is, see what is happening. It is becoming a linear unfolded form and that is getting coated by SDS and it is running. And then the molecular weight marker is going to tell you how big the protein is. Now one very interesting thing about SDS page is this discontinuous gel system. So in SDS page, you guys know that there are two different gels. Okay, so both the gels, they have slight difference in their composition. So what is happening? There is one gel on the top that is called the stacking gel. So in the stacking gel, the pH is maintained at 6.8. Plus it is having larger pores. Now in the buffer, there is glycine, there is chloride ion, and the protein is added. So at this particular pH, glycine is having such a charge that chloride ion, it is obviously the smallest right here. So it is only one ion. So chloride is running the fastest, then the protein is there, and then glycine is there. But then as it enters into the resolving gel, which is having smaller pores, here the pH is 8.8. So here what happens? Glycine is having a slightly different charge, because of which chloride is running and then glycine is running and then the proteins are running and they are getting dissolved. And in the stacking gel, since protein was getting sandwiched between glycinate and chloride ions, it was forming these stacked bands. Okay, so it was uniform migration of the proteins. But in the resolving gel, the proteins are migrating based upon their differences in size. Okay. Now we will look at all of this that we discussed in greater details. Okay. So to improve the resolution, a discontinuous system of stacking and dissolving is used. The stacking gel concentrates all the protein samples into a single band, okay, before entering into the resolving gel where the separation begins. Otherwise, what could happen? As you load the samples, if you start the migration, then the samples that are loaded first will migrate before the samples that are loaded later, right? So the differences between the ionic strength and the pH of the buffers render different properties to the stacking and resolving gel. The former has high porosity and tris chloride buffer of pH 6.8, and the latter has smaller pores and tris chloride buffer of pH 8.8. Okay, so at the stacking gel, the counter ion, that is glycine. So remember, one is called counter ion, the other one is called a trailing ion. Okay, so counter ion or glycine, this remains neutral. It is a Zwitter ion. Because it is a Zwitter ion, it is having almost very low mobility, right? And the chloride ions act as principal current carrier and they migrate towards the anode. In the negatively charged SDS, protein complex are stacked into a thin band between glycine and the chloride ions in the stacking gel. In the resolving gel, what happens is, since the pH has been increased, glycine forms glycinate ions. So glycinate is negatively charged. It is going to migrate faster than the proteins because it is having much less size compared to the proteins. So chloride ions migrate the fastest, then glycinate ions, and finally the proteins. Okay, and that is how they get resolved by the molecular sieving effects. What is molecular sieving? There are these pores, the proteins are migrating through the pores, and sieve is a kind of thing which helps in separation, right? So that is why it is called molecular sieving effect, because the size difference is accounted for over here. Now, protein visualization. Now you see that this gel is there, there are different proteins. Now what do we do? So now we image. Okay, so for visualization, bromophenol blue and xylene cyanol is used to track the progress of the electrophoretic process. Okay, so you can see visually that this process is happening. But then when you just see the gel running, you see only one single band. You don't see these other bands, right, of the proteins. So then from where are they coming? That is used when you use this, they are seen when you use this staining solution. So staining solution has Kumasi brilliant blue in it. It has acetone, it has other different components. Okay, so Kumasi brilliant blue stain and silver staining, which is a lot more sensitive, 100 times more sensitive compared to Kumasi stain, they are used for the final visualization. So Kumasi stain preferably binds to basic residues in a protein such as histidine, lysine and arginine to form a protein dye complex. In the silver staining, silver ions are reduced to metallic silver at sites occupied by the proteins based on the difference in the redox potential. So this is what normally should happen in a gel. You can see that the dye front should be running the fastest and then the proteins should be running. Okay. So different proteins that you can see, they are separated out based on their size, and then you stain them and then you get to see them. Okay? You get to image them as well. Now protein analysis using SDS page. So a stained polyacrylamide gel can be used to assess the purity of the protein and integrity of the protein sample. So if there are more proteins in that lane, that means the protein is not very pure. Right, number one. Second is integrity. So you can see whether that protein has been cleaved or not. A lot of times our proteins, they are not very stable. That is normal. Okay, so what happens is they can get cleaved depending upon the buffer conditions, or whether the protein requires a specific temperature, specific salt conditions. Proteins can get cleaved. Okay, so that also you can see. So this can be done by visually inspecting the bands on a stained gel to determine the number of bands in a given sample, which gives the rough approximation of the number of proteins in the given sample, and whether the bands present are at the expected molecular weight based on the protein standards. Okay, so it helps to estimate the molecular weight of the unknown protein, but this is only an estimation procedure. You will have to go for more sophisticated procedures such as size exclusion chromatography and other techniques, and mass spectrometry to finally conclude if a protein is having a specific mass or not. Okay, and the relative intensity of the bands. One band is thicker, the other one is thinner, so that can tell you whether one is having more protein, the other one is having less protein. Okay, estimate the relative abundance of proteins in that gel. The amount of dye in a band is approximately proportional to the amount of protein in that band. A thick dark protein band indicates a highly concentrated protein, whereas a faint thin band indicates a dilute protein. Okay, so now this is just a small summary slide for you. So if you have a multi-protein complex, you can't tell on an SDS page gel. In the SDS page gel, when you denature, it unfolds the protein and coats them negatively, and they will disrupt all the interactions. But then you can tell that on a native gel. So here all the interactions are maintained. In normal SDS page, you can say how many proteins are there, whether they are dimers, whether they are monomers, that you can study, and you can compare that with the native protein complex, and you can check whether the molecular weight there is kind of matching with the molecular weight here, and from there we can tell the stoichiometry. Okay, so heat and SDS will disrupt all the covalent bonds, but BME has to be added to break the disulfide linkages. Okay, so we will start with the questions. Okay, you can answer in the chat box. Okay, so let's start with the questions. So in two-dimensional page, the correct order of separation is isoelectric focus. First option is SDS page, then isoelectric focusing. And second option is IEF, then SDS page. You can answer in the chat box. Okay, and then we have native page, SDS page, and then we have agarose gel electrophoresis and SDS page. So what is the order of separation? So that is one question. BB. Okay, good. So the purpose of stacking gel is to concentrate proteins into thin bands. Yeah, very good. So we look at the answers quickly. Yes, that is absolutely correct. So in 2D gel electrophoresis, it is first separation horizontally through IEF, then separation vertically through SDS page. And the purpose of stacking gel is something that we've already discussed. So you guys know it. Now we come to the next question. So in SDS page, glycine acts as leading ion, trailing ion, buffer only, detergent. Glycine, okay. B, right, very good. And then we have one numerical. Okay, so in GATE exam, GATE Excel and GATE BT, you can expect a lot of numericals. So these are numerical aptitude tests or NATs. And so in this NAT, they are asking you to calculate in a 50 kDa protein which binds SDS at 1.4 gram SDS per gram of protein. The molecular weight of SDS is given. So they are asking you to calculate the approximate number of SDS molecules bound. Okay. So how are you going to do that? We will look at that now. Okay. Right. So your answer is absolutely correct. Okay. It is a trailing ion. So chloride ion is often the leading ion. Glycine ion is the trailing ion in the stacking gel, and that is what creates the stacking effect. Okay. Now for this particular question, you see 1 gram of protein is binding, so 1 gram of protein binds 1.4 gram of SDS. And for 50 kDa protein, that means how many grams is that, right? So you can see that it is, so the amount of SDS bound is 7, so SDS mass bound is 70 kDa. And so you take that 70 kDa divided by the molecular weight and you get 243 as the right answer. Okay. Right. So now we have another question, another two questions. Which technique preserves the quaternary structure of a protein? Okay. And C. Yeah, very good. Yes. And then we have another question. So there are two proteins, right? So the PI of both the proteins is given. Molecular weight of both the proteins is same. Okay. So in 2D gel, so remember in 2D gel, first they are going to get separated based on PI and then they are going to get separated based on molecular weight. So when it comes to that, there will be no separation. Acha. So you say that it is B. Okay. Let's see. Okay, good. So in 2D gel, they will appear as the same spot, separation in first dimension only, separation in second dimension only, and not separation. Okay, let's look at the right answers. Yes. So it is C and then B. Very good. So you guys are understanding. That is very good. Okay. Now we'll talk about age or agarose gel electrophoresis. So agarose is a linear polymer composed of alternating residues of D-galactose and this 3,6-anhydro L-galactopyrrose. So the chemical formula is given there. The chemical structure, molecular structure is basically given there. And it is extracted from seaweed. So the linkage is also given, alpha 1,3 and beta 1,4 linkage. So these are important because they might ask you questions. In some exams, they ask you all of these questions. So it's important that you remember them. Okay. Now, it is extracted from seaweed or red algae. Agarose forms a gel matrix of agarose molecules which has channels and pores through which the molecules are able to pass. Okay. Now, electrophoretic migration rate of DNA through the agarose gel depends upon various factors such as the size of the DNA molecules, the concentration of the agarose gel, the voltage applied, conformation of DNA, and the buffer used for electrophoresis. So, even the buffer, if there is even small, like something wrong with the buffer, there will be a very drastic change in the gel. Okay, so we need to be careful about everything when it comes to electrophoresis. So the concentration or percentage of agarose defines the pore size of the gel, hence the size of the molecules that pass through. For example, the higher the percentage of agarose, the smaller the pore size, and thus smaller molecules will be able to migrate. Okay. Now, several buffers are used for agarose gel electrophoresis. Most common buffers are TAE buffer and TBE buffer. So tris acetate EDTA and tris borate EDTA. Okay. Now which is used for what cause? So depending upon the size of the molecules that you want to separate, you will either use TAE or TBE buffer. Okay. So the speed of the migration through the gel is also determined through voltage gradient. That is the voltage between the two electrodes. But then when you have very high voltage, then what is going to happen? A lot of resistance is also going to get built up, and that is why the gel will get heated up. And that will actually melt your gel and it will obstruct the migration of the DNA. So it is best to keep the voltage low. Okay. Now there is this something very important. This is a very important question in exams. They will ask you that you have been given a mixture of nucleic acids. All of these nucleic acids are in one vial Now you run this particular gel, and in one lane you will see that there are different bands. Now how are you going to say which band corresponds to what topology of the nucleic acid? Okay, so gel electrophoresis not only separates the DNA based on their size but also based on their topology or the shape. Okay, so what is happening? The DNA molecule that is super coiled, so that is CCC, circular, covalently closed, and it is supercoiled. For example, plasmid DNA, native plasmid DNA, that is going to run the fastest. You take that same DNA, same length, but then you nick one strand. So one strand is cut, the other strand is intact. That is going to run uh, above, right? And then there is something called linear DNA. So you linearize, you cut both the strands, it becomes one linear DNA. So where is that linear DNA going to run? So all of these different things are there. We will see that in the next slide. Okay, right. So now the colorless nucleic acids are tracked during the gel run using a dye, bromophenol blue, loading or tracking dye. And finally, to visualize the DNA, agarose gels are usually stained with ETBr, ethidium bromide. Now, ethidium bromide is once again not very good for us, and that is why a lot of different labs are using other alternatives such as cyber green and all of these different alternatives. Okay, so a fluorescent dye that intercalates between the stacked bases of the nucleotide and fluoresces when excited with ultraviolet light. So you will have to use this ChemiDoc, which is or you can use this visualizer, DNA visualizer instrument. There you can check. Okay, so now applications are size determination, definitely, and then quantity estimation, once again depending upon band intensity, purity assessment, whether there is a single band or more than one band, confirmation of PCR amplification, whether PCR has worked or not, confirmation of molecular cloning, conformation of the DNA and RNA. Now in the next slide we will see this order of migration. Okay, so remember that supercoiled always runs the fastest. Okay, and if there is circular single stranded, so circular single stranded, that means its molecular weight is obviously like much, it is half of the supercoiled DNA which is double stranded, so it is going to run the fastest here in this case, right? Because it is single stranded DNA plus it is closed. Then you have double stranded closed DNA which is circular supercoiled, and then you have covalently bonded, then you have linear DNA, normal linear, and then you have nicked circular at the top. Okay, so nicked circular runs much higher up. So this is an important question which can be expected in your exams. So let's try to solve the MCQs. Okay, so MCQs on SDS page and age. So to visualize protein in an SDS page gel, which of the following staining methods are used? Okay, in the next question you are asked, DNA mobility is inversely proportional to voltage applied, agarose concentration, molecular size or log scale, and buffer ionic strength. Acha, A and C. Good. Let's see the right answer. Yeah. So for the first one, it should be Kumasi staining. That is correct. And for the next one, in agarose gel electrophoresis, DNA mobility is inversely proportional to molecular size or log scale. That is correct. Plus, it is actually also kind of will be affected by agarose concentration. Okay. But in this question, maybe both would be the right answer if that would be an MSQ. But if it is an MCQ, then you will have to choose which is more correct, more appropriate. In the exams, they will ask these confusing questions, okay, where more than one answer can be correct, but you will have to choose the one that is more correct. So if that is an MSQ, both of these would be correct, but if it is an MCQ, only one would be correct. Okay, and if that is the case, then you will have to go for C. Okay. Now, we have another two questions. In agarose gel, the migration of the DNA depends upon. So this is an MSQ. You can answer as many as you want. Size, shape, net charge, agarose concentration, so many different things. Right? And then we have in electrophoresis, DNA will migrate towards. So it is DNA which is what charge? A and D. Let's see. Acha. So we look at the right answer. Okay. So the answer that is provided to us is shape and size of the DNA. In agarose gel, the migration of the DNA depends upon your shape and size of the DNA, and the rate of migration depends upon agarose gel concentration. Okay, so if the agarose gel concentration is very high, then it like the pore size will be smaller, and so the resolution will change based on that. Okay, the rate of migration, so all the DNA molecules will get affected in the same way. And for the second question, it is very straightforward. It is definitely, it's an anion. It is going to migrate towards the anode. Okay. Now, agarose gel electrophoresis is used for which of the following reasons? And the next question is, the pore size of agarose can be reduced by A. Acha. And the next question, AA. Let's see. Yeah, that is very correct. So, agarose gel electrophoresis is used to separate DNA fragments. It can also separate proteins, but it is normally not done. Okay, and since this is like it cannot denature protein, so of course all of the above would not be the right answer. And the pore size of the agarose can be reduced by increasing the agarose concentration. Okay, right. So now we have two more questions. SDS page is used to determine which of the following: pi of the protein, molecular weight of the protein, presence of disulfide bonds in a protein, dissociation constant of a protein ligand complex. B. Right. Very good. And then the next question. What should the next question be? Next answers. B and D. Okay. Let's see. Let's see what's the right answer. Yes. So, SDS page can be used to determine the molecular weight of the protein. That is correct. And it can also determine the presence of disulfide bonds in a protein. Okay, so if there is a disulfide bond in the protein, then let's say which is an intermolecular or intramolecular disulfide bond, depending upon that the mobility of the protein would be different. So if it is an MSQ, you will have to answer mark both of these correct. If it is an MCQ, just only go for molecular weight of the protein. Okay, now the next one. The next one, you see all of these are correct, right? So tris is there in the buffer. Glycine is definitely there in the buffer. Chloride ion is also there because you are adding tris chloride, right? And then SDS is also there, which is helping to give negative charge to the protein. Now we have three more questions. In SDS page, the protein sample is treated with SDS. For what? The next one is, which tracking dye is used in SDS? And the other question is, non-essential component of SDS page is A, B. Okay. Right. So let's look at the right answers. So SDS is majorly used for making the protein negatively charged. It also assists in denaturation of the protein. So the next question is, bromophenol blue is used as a tracking dye. Kumasi brilliant blue is used for staining, right? And non-essential component, what happens if the question is MSQ, right? So if the question is an MSQ, you will also have to answer denaturation of the protein, okay? Because it is a detergent, and detergent helps in denaturation of the protein. So both heat and SDS are helping in denaturation, okay? So if it's an MSQ, then you have to also mark denaturation. Okay. So that is what is happening. So if the protein is to be analyzed, it does not contain cysteine, then there is no need to give BME. Only if there is cysteine in the protein, we have to give. Is it compulsory to answer more than one? Why mercaptoethanol is used if it is non-essential? Achha, so I'll answer your questions one by one. So the first one is it compulsory to answer more than one? So in the MSQs, in GATE, okay, in MSQs, it is written that if the particular question, let's say, there are 2 or 3 answers correct, you are marking only one of them, you will get partial marks, okay. And if the MSQs in that particular MSQ, if let's say you are marking one negative, like you mark extra and that is not correct, so based on that they are going to deduct marks. So all of those instructions will be written in your question paper. So when you practice these questions, you will have to actually check. So it is better to not attempt MSQs if you are not 100% confident, okay. But for MCQs, you guys know it is very simple, it is very direct, right? But yeah, for MSQs, it is best to, if you're not very sure, just mark one that you're very confident about, then at least you'll get partial marks for that. Okay. Yeah. So we'll come to the next question. So next question is, why is BME added if it is not essential? Right. So BME is added only when that a particular protein has disulfide bond or there is any cysteine residue. Okay, so if there is cysteine residue, there is a very high possibility that the protein is forming a dimer. Okay, and because of this disulfide bond, you know that heat denaturation is breaking the covalent bond. SDS is coating it completely, but then this SS bond is not breaking, disulfide bond is not breaking. So if the protein is migrating, it will be migrating at double the molecular weight if it is a if it is forming this like, what do I say, dimer. Acha, what are you saying? If the protein is not characterized, should be used BME. Yeah, so for that, what you have to do is you will have to run 2 different lanes. In 2 different lanes, you will have to run the protein. Okay, so in one of the lane, you can run it with BME. In the other one, you can run it without BME. Without BME and with BME, you will compare. Then you will come to know whether it is forming SS bond or not. So I'll give you an example. Even if the protein is forming intramolecular disulfide bond, and if you are running, you will see a distinct shift in the migration. So because of this disulfide bond, the protein molecule becomes relatively more compact. So it will run faster through the pores. But when there is no disulfide bond, it is a linearized product, you have added BME, it will run higher up compared to this protein with disulfide bond. So there is going to be some shift. Okay. Okay. I hope I have answered your questions. If there are any more questions, you can always ask me. Okay. We'll go to the next slide. So there are 2 more questions. What is the role of BME in SDS page? And then B. Okay, good. To break disulfide bonds. Yes, very good. And the next one is, the pore size of polyacrylamide gel can be changed by changing the acrylamide concentration, changing the bisacrylamide concentration, B and D. Yeah. Okay, good. Very good, both of you. So yes, it is B and D. Correct. Now we go to the next question. So migration of a protein in SDS page is determined by, in SDS page, remember, C is size of the protein, correct. Now what is the role of APS in SDS page or native page? C, let's see, okay. Acha. So to cross-link acrylamide and bisacrylamide, it seems SDS is needed. I will have to check this one. To provide free radicals, is it? That's what we read. Okay, so I'll go through this and I'll tell you in the next class. Okay, if this is if you have to go through it. One second. Yeah. So as we can see here, this APS is helping it to character. Acha. So TEMED is speeding up the decomposition of APS to give a free radical. So the exact role of APS, I will go through it once and I'll check, and then I'll confirm in the next class what the answer of this particular question should be. Okay, whether it should be, right, one second. So whether it should be B or it should be C, I'll confirm in the next class. Okay, so we'll go to the next question. The electrophoretic mobility is the velocity of a macromolecule under a constant electric field. So V divided by E, that is velocity by electric field, right? And mu is equal to, so mu is influenced by what? Charge, mass, shape, all of the above. D, all of the above, right? So now there is an interesting question. So this kind of question you will expect in your exams, okay? So if a protein shows a molecular weight of 100 kDa when it is analyzed by Page and gives a band at 20 kDa, another at 30 kDa, what is the stoichiometry of the protein complex? Okay, DA, right? Yes, so that is correct, D and A. Okay, you guys know how you calculated A. So the equation is given here, very simple way to do it. Now we have a few more questions. So electrophoretic velocity or V of the protein samples dash with increase in voltage, increases, decreases, remains unaffected, may increase or decrease. Then in Lamely buffer of the SDS page, dash is used as loading die. C. C, okay. The next one, B. Bromophenol blue. Yeah, so that is correct. And what about the last one? Glycerol is added to the protein. Achha, so you're saying that it is ABA. Okay, let's see which is the right answer. Achha. So yes, that is correct. ABA is the right answer, because when you increase the voltage, always remember that when you're increasing the voltage, the current is increasing, right? So when the current is increasing, definitely your protein is going to move faster, because this is having the gradient charge separation, that is also increasing, so it is going to move faster. But resistance is also increasing, that is a different story, but then it will definitely move faster. Okay, it will probably not move very uniformly if you increase the voltage too much, but it will move faster. And in Lamely buffer, you know that bromophenol blue is used as loading die. Good. Now glycerol. Why is glycerol added in the Lamely buffer? Why? So glycerol is added to the protein samples before loading them into the wells of the gel. Why? This is because glycerol makes the sample dense and helps them to descend into the well, because your protein is, after all, it is just a solution. It's like watery, very watery, right? So when you're loading it into the gel, how do you know that it is actually going to go in? It might just float away and get dissolved in the buffer. So that is the primary reason this is done. Okay. Now we have pH of stacking gel, dissolving gel, buffer tank. What are the three different pH? Remember stacking gel and dissolving gel pH? And for the next question, BB. Oh, good. Yes. So an SDS molecule binds at the interval of every dash amino acids. Yes, that is correct. 2 amino acids, and the other one is 6.8, 8.8 and 8.3. Okay. We have a few more questions. We have 3 more questions actually. This is there and then 2 others are there, and then we'll end today's class. Okay. Oh, and we will also discuss the previous classes doubt. Okay. So 5 kb plasmid DNA is analyzed on a 1% SDS, 1% agarose gel. 5 kb means your normal vector is normally 5 to 6 kb. Okay. So under denaturing conditions. Now the sample contains, I was telling you about this question, supercoiled DNA, covalently closed supercoiled. There is nicked open circular DNA and there is linear DNA. Okay. Which of the following statements is correct? Supercoiled migrates slowest. Okay. So you are saying that it is C. Right. Right. Yes. So yes, very good. It is C. C is the right answer. Because remember, supercoiled, nicked circular supercoil always travels the fastest. Then will be your linear DNA. So in linear DNA, both the strands are nicked. So it has become linear. But in nicked circular, so one strand is circular, the other one is nicked. Then what happens? It is migrating the slowest. Okay. So that is because agarose gel electrophoresis also takes into account the compactness or the topology or size of the DNA. Okay. Now, one second. Yeah. So this is another question. So a protein of molecular weight 60 forms a homodimer in native conditions. Each monomer has a pi of 5. There are no disulfide bonds. Okay. Now you run an SDS page without disulfide, without BM. Then there is a native page which is run. Acha. So you're saying that it is D. Okay. We'll see. Now there is isoelectric focusing and there is agarose gel electrophoresis. Now what will you see? So in SDS page without beta mercaptoethanol. Considering the question once again, check properly. In native page, SDS page, isoelectric focusing since it's a monomer, and you have it migrates towards something, right? So if its pi is 5 and in agarose gel the buffer is normally 8. TE buffer, TBE buffer, the buffer pH is normally 8.2. Yes. That is the right answer. So B is the right answer. Why? Because in SDS page there is no what is going to happen. I believe both bands will merge in and form a single band. Yes, that is correct. So both the bands will form one single band and the single band will be at 60. Right? And then in native page what is going to happen? The interaction is going to be retained. That is why it is going to be 120 kDa. Then what happens at isoelectric focusing? They are having same pi. They are the same protein. Once again, they will merge together at pH 5. And what about agarose gel electrophoresis? The buffer pH is 8. So what happens? What will be your charge? It will be positive, right? So, no, one second. Okay. This. Yeah, so what happens is the protein is neutral. The protein at neutral pH, that is more than pi, negatively charged, so it migrates towards the anode. Okay, so that is what is going to happen. Now we go to the next one. Now, two proteins A and B, they are having the same molecular weight and same pi. Okay, so this is what we were talking about in the beginning of the class. See, so protein A undergoes phosphorylation at one serine residue. Protein B undergoes glycosylation and this adds 2 kDa. And the sugar moiety is neutral, right? So that means there is no change in charge, but there is change in mass. So what is going to happen? They are analyzed by 2D gel electrophoresis. Which of the following is true? So both shift only in second dimension, both shift only in first dimension. A shifts horizontally, B shifts vertically, A shifts vertically and B shifts horizontally. Okay, let's see. C is the right answer. So both of you are wrong. So why is C the right answer? Because for A, remember phosphorylation is getting added. So there is no change in mass, but then there is change in charge. That is why it is going to show different migration in isoelectric focusing, and that is happening horizontally. So it is going to shift horizontally, right? So A is shifting horizontally, but then when it comes to B, 2 kDa is getting added due to the glycosylation. There is no change in charge. So it is going to not shift at all in the IF, but it is going to shift vertically in SDS, because the one that is having more mass is going to migrate slower. Correct? So that is why C would be the right answer. So the explanation is given to you. Okay. Right, so that is it for today's class. We have all these references. Please go through this virtual labs pages. Agarose electrophoresis and page, both are explained very well here. So if there are any doubts that you have after the class, you can always refer to these references. Okay, that's it for today. And now we will, thank you so much. Now we will go back to the last day's MCQ. Okay, so I'll share that slide. So I'll not share this slide anymore. If there are no more questions, we'll go back to that. And we will share. 1 second. Yeah, so we have lecture 5 here. Right, so we'll go back to that particular question. That we had. Yeah, no, this is not the one. Yeah, so this is the question, right? So we'll look at this question nicely. So see, in this question, they are asking you in size exclusion chromatography, the CEPADEX this thing is given, specification is given, and the column volume is 60 ml. The volume of the stationary phase is 45 ml. So the column volume means the V total, okay, and the volume of the stationary phase is 45 ml. If the elution volume of the solute is 40 ml, we find out the distribution coefficient or KD. Now there are different ways of solving this question. Okay, so now the one that was provided by the NPTEL team, that said that the void volume is equal to 1/3 of the column volume. So that is coming to be around 20 ml. Elution volume is 40, and Vs is 45 ml. So Vs is your volume of the stationary phase. Right, so then Kd is equal to Be minus V naught by Vs, and from there they are getting 0.44. Now there is another way of solving this question, and that is if you take the total column volume or Vt to be 60 ml, then the stationary phase is 45 ml. So then when you subtract these two, then you get this 15 ml, and then Kd is equal to 15 ml, which is your V naught, right? So V naught is 15 ml, and then you get Kd is equal to Be minus V naught by Vt, and this this is actually giving you, not BT, this is your BS. So that is actually giving you 0.55. Now that is also another way of doing. So if that is how you do it, then you wouldn't come closer to any of these answers. Okay, so in that case, both are possibly correct ways of doing it, but then whichever way is giving you the answer, you will have to attempt that, right? So there are sometimes questions like this was taken from the NPTEL previous year's questions, but if there is any such questions in your upcoming exams also, what people do is they normally challenge these questions, right? So if the answer is not very straightforward and if the answer is not close to any of the given answers, then people normally challenge and then they get the marks for that particular question. So everybody gets marks for that particular question. So it is one of those confusing questions. Okay. So you can look into it for better understanding. So what I understood from this question is there are different ways of solving it and that different ways are taking you to different answers. So it's best to understand how to solve a question and then leave it. Okay. Right. Okay. So that's it for today's class. If there are any more doubts, we will discuss otherwise. Acid rain was your topic. Oh, good. Interesting. Okay. How was it? How did it go? Little bit hesitation, but in explanation, but it was good. Yeah. So you will need practice. That is normal. Even in my college days, I was very scared of public speaking. But you'll manage it with time. Okay, all the best to everybody for your work, for everything. We have festivities coming up now. So have a good time. Enjoy Holi, enjoy festivities. Bye bye. See you again. Okay, so we'll end today's class if there are no more doubts. And then we can meet next week once again. Thank you so much. Yeah. Bye bye.